We're interested in learning about how BRCA1 and 2 mutations end up causing cancer in women who have inherited mutations in these genes. And to do that, we're looking at breast and ovarian tumors from patients who are carriers. We've done an experiment where we have looked at all of the genes in these tumors. Using this data, we've been able to look at various signatures that others have shown to be associated with cells that do not have BRCA1 and 2 function from other sorts of model systems. We've looked at our data and we have ended up to find that many of the tumors in BRCA1 and 2 mutation carriers follow a classic pathway. The gene is mutated, another gene called P53 is mutated, and then all of the function of BRCA1 or 2 gets knocked out and a tumor is formed that's very unstable. The genome is very unstable, lots of chromosome breaks. Those tumors are very responsive to platinum chemotherapy and PARP inhibitors. Our early data has shown by looking at BRCA1 and 2 mutant cancers that some cancers actually don't seem to follow that classic pathway of formation. These tumors seem to not have mutations in the gene P53. They don't always look like they're losing all of their BRCA1 and 2 function. And importantly, we've seen in these patients that they do not appear to respond as well to certain targeted therapies like platinum-based chemotherapy. So we're very interested in expanding this and trying to understand this more. Why do these women who are carrying BRCA1 and 2 mutations get the tumors that form along this potential, what I'm calling an alternative pathway? And by studying more tumors from these patients, including expanding into other tumor types, can we start to understand, could this be a mechanism of what's called primary resistance, where tumors actually do not respond to, say, a platinum chemotherapy or a PARP inhibitor? And could we consider assaying these sorts of functions of BRCA1 and 2 in these tumors to help us determine which patients may respond or not respond? And so that is one potential implication of this work. And in addition, we'd like to expand our work into usage of cell model systems or potentially other model systems to try to understand, how do these tumors even form in the first place? Are they still related to the mutation that the woman or man carries, or are they coincidental? And we think that this is, really important, one, from a basic understanding of BRCA1 and 2 biology, but also to understand how best to treat these patients who have these mutations. As a young investigator in science, it is getting increasingly harder to obtain funding from government sources. And it's a really critical time point in our careers. For many of us, we've been in training for a number of years, and the draw to leave research is very, very great. And if one doesn't obtain funding, usually that's going to be when they stop doing research, to be perfectly honest. One of the sources of funding that I've gotten is through a philanthropic donation, and that has been absolutely critical in helping to protect my time so that I can spend time doing research. I still do have clinical activities as a medical oncologist, but I really want to be able to focus my time on the research. And really the way that I can do that is being protected with the funding that I'm getting through the philanthropic donation from the Connor family and through the Vassar Center. What this is allowing me to do is to get the preliminary data that I need to be competitive for a large grant from the NIH. And so actually right now I'm putting in a career development award based on the data that I generated from the philanthropic donation. And really that grant is a 5-year grant. It'll really launch my career as an independent scientist. And so I really couldn't be doing what I'm doing without this philanthropic support. It's absolutely really critical. The most important impact that the Vassar Center has is bringing together people from different fields. And so in science in general, I think that people have been very siloed, and there's basic scientists and there's clinical scientists, and a lot of times there's not a lot of crosstalk. They just don't go to the same meetings. They don't interact, really. And if you don't interact, you're not going to collaborate. And as somebody like me, who I'm trained in basic science, I have a PhD in molecular and cellular biology, but I'm also a medical oncologist, I can see sort of both fields, but I can see how there's a push to one versus the other. And it really shouldn't be that way. We really should be all sort of interacting together and getting ideas from each other, because we learn so much more by interacting with each other. And so with the Bassler Center, I think one of its major impacts has been through having monthly conferences where the clinical and the basic science people interact. Having a conference like today where we're seeing all sides of the research that are important to BRCA 1 and 2 biology. And I think that that's one of the major impacts that this will have. Number two, I think that another major impact that the center has on a public health domain is awareness, of course. And I think that, for a very long time, potentially there was some stigma associated with having genetic mutations. And by increasing awareness that this is a common thing, that is something that we should embrace and be able to take power over, that is another place that the Bassler Center has a huge impact. I think that the Bassler Center can have a huge impact on sort of in general how we think about risk to cancer, getting patients to talk about it, getting patients to talk to their physicians about it, and really being able to take control of their risk.